Hello, fellow Reptile Genetics addicts. Welcome to episode 9 of Reptile Genetics Weekly. Very happy to be here with you live again, our second time doing a live show. And as always, we'll start out with an update on samples and results. So right now, the samples that we are prepping and have a run going are the ones that came in by May 30th. So if you have a shed received by date of May 30th or earlier, and you're looking for results, those will likely come out on Monday or Tuesday. So just a few days out. And then about a week and a half after that, we'll have the next batch. So lots coming through. We appreciate it. All these samples coming in are helping us to fund designing new ones, which we'll get to talk about today, which is fun and exciting. Kayla, how are you doing today?
I'm doing well. Here, hang on. I gotta make sure. Okay, it's gonna surprise us anyway. I'm doing great. How are you?
And we have a third today.
A wild Sean appears.
Hello.
So many of you have talked to Sean, whether it's messaging or on the phone, and so we wanted to get Sean on here, especially because we got fun stuff to talk about today. So Sean, well, I guess we've talked about this a little bit before, but Sean and I have been friends. It was back in the day before social media. Really, we were in forums online, and especially the Morrill of Pythons forum. But in 2011 at the Tinley Reptile Show, we ended up joining forces as far as me I just gotten my PhD in genetics, doing a ball python genetics dissertation, and Sean's serial business starter and owner. And so he spread that disease to my brain a little bit, and I've been thinking about it every day since then, 2011. So
a few things happened, and now we're here.
Yeah. So, many people don't know that. It seems like all of a sudden we have all this cool stuff, but Sean and I have been chipping away at this for over 10 years.
Yeah. That's awesome. So something Sean was mentioning something about, he was also had a little TV fame. He was on a show called Alone, I think. Something like that.
Alone the Beast.
One of the spinoffs of Alone, a show called Alone the Beast. I was in episode 6 called Tainted Water. I think it's free on Hulu. It's a couple dollars on Amazon or somewhere, I guess. But that's cool. I spent a lot of time outside, and my love of reptiles obviously spawns from there. But I also enjoy hunting, and I don't know. I found myself invited to participate in a reality TV show, and I made it to the end. It was kind of fun.
Very nice.
You get to see Sean under some stress and see how he reacts.
Yeah.
I think I did okay.
Now he just does it behind the scenes. So a lot of y'all who have who have done like email correspondence and phone correspondence too with us have possibly also interacted with Sean. So
a lot of the generic RGI replies are coming from me. Yeah.
Those are important.
Is the chief garbage taker outer, as he's labeled himself. So
yeah.
Sorry,
go ahead.
Truth be told, this 100% would not be where it is without Sean having the kind of faith and being able to spread the ability to take on. For me, it's scary to let go. I had a very nice, easy corporate pharmaceutical job where I was doing research and development, doing genetic tests. But to go out on a limb and be on my own, that would be way too scary for me. So this is definitely made possible because Sean was able to help me realize, that it's something that I love doing, and he loves as well. And so let's make it happen. So we did.
It's awesome.
Keep sending jets in, keep us busy so Dr. Ben can stay employed.
There you go. And me.
Yes. So I'll still be happy even if I start running out of money, but my wife won't be. So it's mostly keep having money coming in to pay for everything and the 4 kids. So, but yeah, we're very thankful for all of you, all the sheds and samples and tests that have been coming. It's awesome.
Mm-hmm. Well, we have 25 people in here right now, actually. So hi, everybody. And see, we got comments coming in. from everywhere. Grays, reptiles, Terry, high desert pythons, straight blast pythons, Andrew Rose, Lori Torini, Logan the Bean Master. That's an amazing name.
That's my
Just
son.
you guys. That's your son.
Yep.
I appreciate Logan's sense of humor. That's
That's Logan. And we do have several on Instagram as well. So those of you on Instagram, sorry, you're only seeing me. If you want to get on YouTube or Facebook, you'll be able to see Kayla and Sean as well. But otherwise, I'm getting lots of waves on the Instagram feed. So yeah, thank you all for joining. And like I said, feel free to switch over if you want to see everybody. Otherwise, you should be able to hear everything and get to see me and my facial expressions. But yeah, I guess we should just go ahead and jump into the happy news.
Let's get on with
it. So, well, I guess what we could do is say the hint for, so we've got the genetic stripe test going, which many, if not all of you know that because Clutch made that available. Mm-hmm. And it will be available soon on Morph Market as well. But the other two tests, we have two more new ones to announce. And the thumbnail for this show, which is also in our Instagram and our Facebook, that animal has both the genes that this new test is for. So maybe what we'll do is we'll just say that and let you all take a look at the thumbnail for 5 or 10 minutes while we answer a couple of questions and see if anyone can guess.
Yeah, let me pull it up real quick.
Oh yeah, I guess you can just show it on the screen, huh?
Yeah. See, I figured that'll make things a little easier for you guys.
While you're doing that, we do have a question on Instagram. It says, do you have a test for blade planned? I haven't started focusing on that yet, but we certainly can. I would say that's not likely to happen in the next 3 or 4 months. But eventually we can work towards that. Yeah, that's another interesting one. So yeah, this animal here has both the genes that this test is for, and I guess I'll give you another hint in that it's not neither of them are recessive genes, because that would be kind of cheating to show you a double het that has the genes. So these genes are expressed in this animal that we're testing for. So kind of fun, a little bit different. A lot of the ones we've focused on have been recessive genes, but these two new ones are not.
Yeah, this is going to be a surprise for me too, because I don't know what's causing, like, I'm colubrid people. I don't know what's causing that crazy pixelation and all of that. So you guys are finding out when I do.
All right.
Yeah. Are we still teasing? Are we doing more questions?
Yeah. Why don't you bring up, you had a couple of comments that went up before we started the show.
Yeah. Well, okay, first one, this one's not a question. It just makes me laugh. It's Terry Pruitt. He says he's been holding out and is going to hit us with 10 new morph tests.
That is actually a pretty good comment, because in the month of June, it's possible we could have 10. So we've got 3 this week, and there's another 3, 4, 5 more that are very close. That I would be surprised if we don't get all of them by the end of June. And maybe even a couple more after that. So yeah, today we're only hitting you with 3, which, one, the cat's already out of the bag on G-Stripe, but yeah, by the end of June, it could easily be 10.
Gosh, that would be crazy. So awesome.
Lots of research going on.
Oh yeah. And it's all because of you guys sending in those sheds and telling your friends to send in sheds. Actually, this one is from High Desert Pythons, and it's about something they've been doing, sending in sperm casings from a couple of their males. I don't know if y'all have had a chance to run that, but I thought it was cool.
So we have. So I've purposely not talked about this publicly as a way to get samples, just because the number one potential problem that we would run into if you're going to use sperm plugs or sperm casings this is nice, because if it's a mature male, you don't have to wait for him to shed. You can do kind of like you're going to pop to find out if it's a male or a female, not with as much force, but just do a bit of a pop. And if a sperm plug or part of a sperm casing comes out, you can collect that, let that dry out completely, put it in a bag, a Ziploc bag, and just circle it with a magic marker. So it's a little bit faster for us that are doing the sampling. Go ahead and circle that. And that does work. But the number one thing you need to worry about, and the reason why I haven't talked about it publicly, is if that male has been in with a female. So if he's like a posset clown and he's been in with a clown female, then, there could be some cross contamination. It might come out saying he's a het, but it's really the clown, DNA from the female. So you have to be really careful if you're going to use sperm plugs. Make sure that male hasn't been in with any other animals for a while. That's what would concern me. But yeah, it works beautifully. We've done it many times and it's worked well every time.
That's awesome. I didn't realize. Actually, no, it makes sense that you could do that, but hasn't something I'd considered. And sorry for springing that publicly on you if you didn't mean to do that.
No worries. It's fine. People just have to make sure. And we're talking to the genetics addicts in this show. So
yeah,
these people care. So yeah, just be mindful of that. One other thing I'll throw in with that is, there's one other thing I was going to say. Mm-hmm. I don't remember now.
Before the sperm casings, before
it's all right.
We'll remember eventually. Let's see. Next we have. Let's see. Was this the question you answered? The question about hurricane, trick, and blitz, right?
I don't think so.
Oh, okay. Well, Gray's Reptiles also asks, is RGI doing any testing with hurricane, trick, and blitz?
So the most sheds I have right now of those three are hurricane. So I have started collecting those. I don't have enough to start doing the sequencing yet. So if people have hurricane animals, I definitely could use more sheds. I'm pretty much not going to start testing until I have at least 20. And if I can get 40 to 50, that's ideal. So I'm kind of in the less than 10, I think, maybe 10.
All right.
So get those sheds coming in.
Yes. Let's see. We've got people excited for Desert Ghost. We have people that are. Oh, here's one from Jaffe Tesfa. I really hope I'm pronouncing your name right. When do you think you will have the Monarch test, and do you expect it to be the same as Ultramel?
So we will have Monarch test probably in a couple months. So that one is still in sequencing, so it will take a little bit of time. So far, the Ultramel test, and we did actually have. If you don't mind, Kayla, why don't you bring up the Ultramel question that we had that came in recently?
Not a problem. One sec.
So we'll kind of answer both these questions at once.
Mm-hmm.
So I had seen a posting saying that Ultramel will be offered as a testable morph soon, but have not seen any kind of timeline update on it. Is there any kind of ETA on that gene? I have a couple of sheds I would like to send in for confirmation. That's from Andrea via an email. So Ultramel, and Sean and I have been talking about this a lot lately because this has popped up in multiple ball python morphs, which as we branch out into other species, I'm sure we'll see it there as well. But Ultramel didn't end up being as simple as we thought. There's at least 2 different mutations that cause Ultramel. And we're still assuming that they're in the same gene, but both Dr. Seidel and myself, and there's actually 1 or 2 other groups that may also be looking. And so far, as far as I know, nobody's found that other mutation. I've referred to them as Ultramel A and Ultramel B. So Ultramel A is the more common mutation, and that's the one we can test for. So some people have said, hey, I know you can't test for both, but I'd still like to test for Ultramel A. And so we worked out a way, message us if you want to do that. But if we know that like say you have a het Ultramel combo male that you're breeding, if we test him and find out that he's Ultramel A, then we know that any offspring from him will be able to test as possets because they would have the one we can test for. So anyway, Ultramel is just a little more complicated. Albino is the same way, but we did figure out how to do albino, all of the different alleles that make an albino or candy toffee. We can test for all of them. Ultramel, we can only test for one of the at least 2, and there could be more, but more than likely it's 2. So now coming back to Monarch, with Monarch, so far I know that Monarch is not the same as Ultramel A. So that when I run Ultramel on Monarch samples, the Ultramel A test, it's always negative. What I don't know is what Ultramel B is. So I can't say for sure that it's 100% different than an Ultramel, but I can tell you that in a month, I will know which chromosome Monarch is on, even if I don't have a test yet. And I would at least know if they're on the same chromosome and the same, general location, or if they're on completely separate chromosomes. So we'll be able to announce that before we have the actual test ready. And so we'll definitely make that information available as soon as we have it.
Very good. We've got another question from Scales and Tails Breeding that says, will there ever be a test to determine if a banana/coral glow is a female maker or male maker?
Yep. Yeah, we'll be able to do that pretty soon. I would say that's sometime this summer. So we actually are putting together a really cool scientific paper. It's going to take some time. It's a pretty big project with many people that we're collaborating with. But it will talk specifically about banana/coral glow and the sex determination gene in Python, or at least ball pythons. So yeah, we will have a test that will be able to tell you that.
Awesome.
Here's one that.
There we go.
Yeah, here we go. So here's another question. This is a good question that we definitely want to hit. So Critical Constrictors ask, does all the gene tests at the current price be applicable to the current test plus the 10 new tests? So as of right now, yes. So these 3 that are getting added, the price will not go up. So you would go from 11 to 14 genes, and it's going to still be the same price. Sean, maybe it'd be a good time for you to jump in where you think that is going once we get up to 20, once we get up to 30.
It's important for people to understand that the more mutations we wrap up in a panel, we have to have them work together, and they all have a different sets of primers that can work together. But the more of these mutations that we have in one test, the more these primers want to counteract each other, if that makes sense. So at some point, we will probably try and split panels into a couple of different panels. If and when we do that, there'll probably be an accompanying price drop on those, particularly as we have other products coming to market. We have some pretty neat stuff that we want to bring to market here pretty soon. That will probably be a better option than the panel, and we'll probably talk about that in the coming month or 6 weeks in further episodes. But for right now, it's just really important for people to understand that at some point we have to stop, because we can't make everything work together the way we're doing it right now. But that doesn't mean there won't be an offering in the future that you won't be able to do that. But the way we're doing panels right now, at some point we're probably going to have to limit it, but not yet. So far, these next 3 should multiplex just fine. So.
Yep. And even 20, that shouldn't be a problem. Once we get to 30, it might be kind of a pain, but 20 should be no problem. And, some potential ways to split it, maybe we have like a yellow belly complex panel, we have a recessive panel. I think those are some specific groups of genes people would want to test for, and not necessarily need, 30 different tests. They maybe just want to know, is my animal het for anything? So they want to run the recessive panel, or is it, I think it looks like
leucistic testing and things like that. Leucistic,
what was that?
I said like a potential for leucistic panels, things like that.
Yeah. Blue-eyed Lucy panel, black-eyed Lucy panel. Yeah. Yep.
Can you guys still hear me, just to make sure?
Yep.
Okay, good. It looks like my camera's frozen, so I'm perpetually in thinking face. But that's fine. I'll just stop my cam for now. So actually related question just above that one from Facebook. This one's from Andrew Rose. Is the full panel going to, is it going to cost? Will that have to increase to cover the total number of tests over time?
So our plan is, it's actually because of all of you, we can keep the price the same, even though more tests are coming. So as we have more and more volume coming, you all are having more and more come. And as we have more tests available, I know some people were waiting till Ghost was available. and then they're sending in POS double heads, and now they're waiting for G-stripe now that that's here. So we're getting more and more orders coming in. So as more orders come in, the volume goes up, and that makes it so we can keep the price the same. So even when it gets up to 20, I would imagine we can still keep it at $120, even though you're going to get 20 tests because of you all, because of that volume. When it comes to sequencing and analysis, the name of the game is volume. And so that's kind of my main job, is to be a liaison between us reptile nerds and the DNA nerds, and try to make it so they realize, oh, there's a lot of these people. We should make them a priority and give them a good price. So that's what I'm trying to do, is get enough volume going so they're like, oh, these are legitimate customers that keep having stuff coming through. Let's give them a better price. And so yeah, I would expect up to 20, maybe even up to 25, that price can stay the same. Once we get around 25 and especially to 30, that's when we probably will need to break up the tests. There's another type of test that we're working on that will be a pretty cool kind of whole genome thing. And so we may, kind of switch it to that for people that want to test for everything that we have. And then we would have panels that would be kind of the 5 to 15 genes per panel. But yeah, for now, as we get up to 2025, I see that price being the same. Is that what you think, Sean?
Yeah, I think so. I mentioned it once before. At some point, we're going to probably come down on the panel price when we release some of the other offerings. That said, we don't really know how we want to break it up yet. And however we decide to do it, because we have two vendors, Clutch and Morph Market, the two best players in the industry by far. If we ask for a change, they both have to be able to do it. So it requires a software change for them to change prices, change how right now when someone orders a panel, it just includes all of the mutations. And when we ask them that we want to change that requires a software change for them to go, okay, so now it's a different product offering. It doesn't just include every mutation anymore. So it's a little bit time consuming and complicated. I wouldn't expect any of these changes to happen until we get our first year behind us, which will be sometime October, November, and then probably slow season, January, February, March, when it's real cold and not a lot of people are shipping snakes, is when we'll be doing a lot of software changes, and you'll start to see some of those things happen.
Yeah.
All right.
And the Root Ball Exotics here is kind of giving a example of maybe an a la carte type of offering where you can pick 5 morphs or 10 morphs or 15. So the price is for 5 or 10 or 15, and then they can choose. So that would go back to what Sean was just saying. We would have to go to Morph Market and Clutch and say, hey, can you guys code this way so we can make this offering as like an a la carte type of a thing? And that's possible. And if that's what more people want, then we can certainly try to make that happen. I would imagine that'll be harder to code than if we have like a set, recessive panel and yellow belly panel. But, we'll see as time goes on.
Good stuff. What else we got? We have this one's easy answer. From Jamie Rising Phoenix Reptiles. Are you guys doing puzzle?
Yes. Yeah, that one is at the same point as Monarch. So it's in sequencing. Also VPI Xanthic and TSK Xanthic. All 4 of those are in sequencing right now. And so I expect to have something probably in a couple months. So by the end of the summer, even if we have some sticking points that we need to work through, by the end of the summer, we should have a pretty good shot at most, if not all 4, 2 or 3 of those, maybe all 4 of those by the end of the summer.
Very good. Figured I'd throw this picture back up because we got a bunch of guesses on it. Let's see. Question from Samuel Ceresi from Facebook. Is high OD the same as regular OD?
So that's something I've talked with people about, getting specific samples in. The most difficult part is having samples that I know for sure are high OD or super high OD, and then samples that I know are not OD, not super, or not high OD or super high OD, to be able to compare. So getting the proper samples may be a little complicated, but that's something I've worked with lots of Orange Dream. It's something I definitely am interested in playing with.
All right. Let's see. Another person's asking, this is BBM Reptiles. I don't know if this was asked already, but is Monsoon on the list of tests y'all are working on?
Yeah, so that's one that I am still collecting sheds for, but I think that on the next, so Monsoon and TSK, the 2A Xanthics and Monarch, those are kind of in the summer. Those will come out. Monsoon is in the next batch after that. So sometime in the fall, we should be able to get enough sheds and have something good on Monsoon.
All right, thank you. Another is, this is from High Desert Pythons. I need some RGI merch.
The three of us have actually talked about that.
Working on that, actually. Finding the right merch company for us. And actually, we want to know, what sort of merch do you guys want? Is it shirts? Is it hats? Is it something else? Sound off in the comments. Let us know.
Yes. Yeah. The little bit that we did, we took to some reptile shows and handed out. Sean made that happen. So yeah, we'll see if we can get that rolling before too long.
All right. Yeah, and we got some people in the comments. They want hats and shirts. Cool. I could see people wanting like stickers to put on their racks or bins or something like that. I would do that.
Can I say something really quick?
Yeah.
Of course.
Just a personal note. Everybody that when you're sending in your sheds, keep sending me more stickers, because every time I get a new one, I'm slapping them up on the wall in the RGI lab. And when we get enough of them, I'll take a picture for everybody to see.
Sean, do you have a picture of the? Well, do you have a working picture, or do you want to wait till it's like?
I don't have a working picture yet, but it was just something I thought of the other day, because every once in a while someone will send us a handful of stickers along with their sheds, and I've been just slapping them up on this wall. And the other day I was like, I ought to just keep doing that. When I've got 30, 40, or 50 of them, it'd be cool just to take a picture of it and show everybody that we have an awful lot of people that are helping us out here, and to be able to thank everybody just by showing them something like that would be pretty cool.
Yeah. Will's Hella Heat says he'll send you a sticker book as soon as he gets puzzle testing.
All right, we're on it. I'll take you up on that.
Got it.
All right, Kayla, let's get that picture back up and let people know. All right, so this animal, I haven't been following the comments very well, so I don't know what guesses there have been. But this animal is an Orange Dream Black Pastel Enchi Calico. And possibly yellow belly. I don't think that it is. Actually, this is one that I have since sold. Otherwise, I would have tested it for yellow belly by now. But so yeah, the two new tests are Enchi and Black Pastel. So we're pretty excited about that. Something a little different. That's not a recessive, but it is something that with all the multi-gene animals you all are producing, sometimes that's something that can hide. So we're pretty excited to have that available. I know me personally, a lot of the time, just because of the animals I'm breeding, my question is whether it's a super Enchi or an Enchi. And some combos, it's hard for me to tell the difference. But yeah, so we've got that going. And for people that have mystery animals that are white snakes or 6 gene plus animals, and you want to know if Black Pastel or Enchi are in there, we can tell you now.
Awesome. That is great, and those are also very popular morphs, so that's fantastic.
Yes.
Awesome. So glad to drop those. Do we want to do more Q&As?
Yeah, let's do it.
Okay. Sounds good. Pulling up comments again. Oh, this one's from Will's Hella Heat. How many individual tests does it take to make it worth you guys for sequencing a gene?
Right now, we, a lot of this is Sean's focus, ability to help make this happen quickly. Right now, we are dedicating a lot of money. I can tell you, hopefully Sean doesn't get mad at me for saying this, but RGI as a business is not making money. We are putting more money into research and development than is coming in.
We're not making money yet, but that's normal for a young business.
Yes, and it is because we are just turning that around and putting it right into research and development. because
Yeah.
we want to make lots more things available for you all.
Literally every spare penny we have is going into trying to develop new tests, especially right now. We're pushing really, really hard. In fact, we were going to talk a little bit about some other stuff that we think is really close before we end the show today. So that may be one of the final things we talk about, just to keep you all hanging out for a little bit longer.
Yes.
All right. Another one from Facebook. This is Mary Hubbard, and she's asking, are you working on paint or sentinel?
We have a few paint and sentinel samples, but not enough. So that's one I need more sheds.
Mhm.
To finish that other question, so he was asking how many do we need to make it worth it?
Oh, yeah.
So right now, sorry, I go on tangents. Sean knows this really well after all these years.
Me too. It's okay.
Most of the time I can connect it back.
Yeah.
So right now, for these next few months, we're dedicating a lot of research and development money and time. And so right now, if you have enough of let's say it's a brand new mutation, send me some pictures and show me. You're going to have at least 20 individuals that have that mutation, that morph, for us to really have a prayer of being able to pick it out. So it's not necessarily right now whether it's worth it or not. Now, a year from now, that might be different. A year from now, we might have, 50 or 60 ball python morphs, and we're off doing something, with corn snakes and hognoses and retics and Burmeses, and you come to me and say, hey, we want this new ball python morph. That might be different. We might say, hey, we need to have this or that to be able to do that. Go ahead, Sean.
I want to say something, because I may be, but when it comes to the number of sheds that we need, homozygous is way more valuable than het when we're trying to develop a new test, right? So I think, and Ben can correct me if I'm wrong, we'd much rather have 20 homozygous animals than 40 het. The homozygous helps us way more. We can still use the hets, absolutely, but the homozygous is what helps us get there. And the second part of that is, whenever you guys are sending sheds in, both platforms give you the opportunity to list the other mutations that animal has. It's really important that you fill those out for us, because we save all that stuff. When it comes in and you have a mutation in an animal that we want to develop a test for, even though you got your test results months ago, we still keep your shed because we're going to use it on a research run in the future. So please, please, please tell us what your animal that has got in it, particularly if it's a homozygous representation of that mutation.
Yeah, like when I was sending off all those monarch and puzzle sheds, I was digging back into stuff that people sent us, 6-8 months ago. And the information that Sean saved as he's brought sheds in, I can look them up, and we have them categorized from when those sheds came in. So I can go directly to a box. I pull that box out, go to that bag, and I can just look through this bag of a few sheds and find the one that's, number blah, blah, blah. And I know that's a monarch, or that's
it's like a giant Dewey Decimal System for snake sheds over here.
Yeah.
I told you, we're reptile genetics addicts. This is what we do. At one of these days, I should just take a picture of all of the various, I'm like a reptile shed squirrel. I squirrel away all the places that my wife doesn't get mad at me for. I've got these boxes from when we moved that are like 3 foot by 3 foot by 2 foot. I've got like 5 or 6 of them jam packed full.
If I had room at my apartment, I'd store them here too, but
well, it's only going to get worse as we break into other mutations, corn snakes,
oh yeah,
mineral. I mean, we're going to keep all of those sheds too. So those of you that aren't ball python folks that are interested in a lot of the other species, we do the same thing. When you send in that, sex determination test on your corn snake, I really want to know what's in it, because we're saving those sheds for future research.
Yeah.
Sorry, I was trying to pull something up, but I guess it made something else pull up. You got this going. I shouldn't mess with it, huh?
Oh no, it's okay. You can. I mean, hey, rainbow is one outside of puzzle. I really wanted to ask. I mean, that's okay. That's
yeah.
Cool.
So rainbow, I have very few sheds. I probably have. That's another one I have less than 10 of. So if you yourself or know someone that has rainbow sheds, send them to me. As soon as I get 20 to 30, I'll get that going.
Can I say something else also, by the way? And I know we're not talking to the entire hobby here, but I do know that a lot of people have expressed to me in person at shows that they're uncomfortable registering their animals with all the mutations because there are other eyes on the software, and I totally get that, but nobody sees the bags but me. So when the bags come in and they're labeled with a particular mutation, I take particular note. So if you have rainbow, if you have any of these other mutations, lace and things that we don't have a lot of samples for, if you don't want to put it in the system, write it on the bag. I'll still see it and I'll get it. And we need them. The faster we get those, the more tests we can work on.
Mm-hmm. That's good to know. And probably something, I don't think that's something we've addressed here either, like on our live streams or videos. So that's good. We were talking about other species that aren't just ball pythons. So Reptile Exotics is asking, how many more sheds do you need before the first hognose morph will be available? Probably guessing it would probably be Conda or albino.
So the biggest difficulty with doing a bunch of hognose morphs is our hands are going to be tied a little bit at first to morphs that we are confident are similar to what are in ball pythons, because for us to have all of the genomic information to make comparisons for hognoses, we need to do a really expensive, in-depth, like we're talking tens of thousands of dollars to sequence the hognose genome if somebody else doesn't beat us to it, which would be fine with us. If somebody else does beat us to it, then we don't have to pay for it. But
yeah, sharing is caring.
I know there are people that have talked about doing it. That would be awesome if somebody does. If no one beats us to it, we may pay that. But so like with albino, we can do that because albino has been found in ball pythons. We know the genetics in humans and mice and all kinds. So we know that the few places we can narrow down 1.5 billion base pairs down to just a few thousand, which still sounds like a lot, but 20 to 100,000 is way better than 1.5 billion. So we can go ahead and in species we don't have a really good reference or whole genome, a really deep one, with morphs that are likely to be the same in the ball python or other species that have been identified. But something like Conda, unless somebody knows what maybe an equivalent mutation would be in a ball python, we wouldn't know where to look very easily. But the good thing is, if we start out with morphs like albino and get some sequencing going, then we'll have somewhat of a scaffold to work from for mutations that may be hognose specific. So it'll take some time. Once again, it just takes sheds. So with the ball python, nothing really happened until I had 400 or 500 sheds. And then that's when I was able to start doing things. So we just need these sheds coming in.
Yep. Tell all of your breeder friends, the guy who bred your snake, like that's how it's going to happen. Speaking as someone who also keeps hognose snakes and would love to see that. Lavender especially, that's my favorite. Going back to ball pythons, we have from Critical Constrictors, with Enchi and black pastel being allelic, how does that look when genotyping?
So that's a very thoughtful question. I like it. So some people aren't even aware that they are allelic, but they are. So that's another cool thing about this. If you get it tested for both black pastel and Enchi, what you would get is a result of either negative, so it doesn't have any allele for black pastel or any allele for Enchi. And then if the animal was both black pastel and Enchi, you would get a het for both. And if it was a super Enchi, you get homozygous. If it was a super black pastel, you get homozygous as your result. And then what you would never get is a homozygous Enchi black pastel, because that's not possible since they're allelic. So you'd either have one copy of each or two copies of one or the other. You'd never have two copies of both. But the test is unique. It's a different mutation, different marker for black pastel than it is for Enchi. So we can tell the difference. So we know if that animal has one copy of Enchi or one copy of black pastel or one of each or none of either. We can tell all of that from these two tests.
Very good. Another one from Kavik River Exotics from Facebook. What would be your projected time frame for a panel to be available for all the recessive genes? Like if you were doing a recessive ball python morph panel.
Any of you
if you decided to do that.
Any of you that have followed what we've done so far, it's very difficult to predict the future. Like we talked a little bit earlier about ultramel and albino being more complicated than anyone knew to begin with, because there were multiple mutations. And actually yellow belly is the same way. There are 2 different mutations in the same gene that cause yellow belly, and they look identical. Us as breeders and keepers, you have one that's the -17 yellow belly and one that's the -1 yellow belly. They look exactly the same, but as a genetics, looking at the DNA, I can tell the difference between those 2 animals. So it's more complicated than we can see on the outside. And so, to be able to tell you for sure when we could do a recessive panel, I would say it's likely that around the time that we have like 30 or more, that would be maybe when we would start doing like or at least 25 to 30. That's when we might break out and have a yellow belly panel, a recessive panel. And it would just be of the ones we have to that point. I don't know that we'll be able to get all of the recessives. It'll take some time because some of them are pretty new and there just aren't very many individuals. And like we said earlier, we have to have 20 to 50 individuals to really start down that path. So yeah, I would say hopefully within the next year we have the 30 or more and mutations, and we're able to break out and have smaller panels. What do you think, Sean?
Yeah, I think that's right. The plan has been with both of the vendors, at some point after year 1, we need to talk about how we're going to start breaking some of this apart. Obviously, if we have 50 recessive mutations or 50 recessive genes, that's a lot. We may not be able to run all of them, right? And how many known ball python morphs are there now? That we suspect 150-ish to be safe.
Oh, geez.
Right. Yeah. At some point, we're going to get to a magic number. Let's just call it mutations for ball pythons, and that number is 50. We're going to slow down and start focusing on other species, because the colubrid folks and the boa folks and other species of python, they've got requests in with us too, and we can't start on those until we slow down a little bit with ball pythons. Obviously, ball pythons is the biggest market by far, so we want to service that as best we can. However, we're going to get to a magic number, and we don't know what that is yet, where we have to kind of put the brakes on a little bit and start spending, okay, the next 3 months of research is going to be dedicated to carpet pythons or corn snakes or whatever. So we can, keep everybody in the hobby just as interested. But tell you sure, the panels are going to be broken up, and I can't tell you for sure if we'll ever be able to do all the recessives at one time, because it's a matter of them working together. Sometimes they just don't work well together. So the answer is maybe, but we also probably will never have all of the recessive genes all at one time. So yeah, it's a really difficult question to answer for multiple reasons, I guess.
But we're definitely on it and thinking about it every day. There's a couple of questions from Snake Morphs 1 on Instagram. He said, and cinnamon 2 ? He said cinnamon is allelic with black pastel and enchi, right? So yeah, that's right. Cinnamon is allelic with black pastel and enchi. Honestly, the runs I've done so far, I've focused on running black pastel and enchi, and then animals I know did not have black pastel or enchi when I've done the testing for these tests. It's the validation of these tests. So honestly, I haven't run any cinnamons, so I'll do that. My guess is it's going to be different enough that on this test for enchi or black pastel, a cinnamon would come up negative, but I won't know that till I run them. So that's a good point and makes obvious sense. I should have thrown 4 or 5 of those on there just to see, but I honestly just didn't think about it because I was focused on getting these 3 ready to make available. But yeah, I'll try to do that on this next run. I'll throw a few cinnamons or super cinnamons on from the sheds you all have sent in and see how that comes out on the black pastel and enchi tests.
Awesome. Next we have from Windy City Morphs. Is cryptic in the list of genes that you're working on?
So that's actually one that some of you may know from watching last week. I said we have a chance for 5, and that's one of them I was hoping I would get this week, but it didn't work. So,
oh man, so close.
Yeah, that one was a complete fail. So I'm glad the others worked. They weren't all like cryptic. Cryptic bombed me real bad. So I don't know how I missed so badly on that one this time, but I do have some other candidates to try for cryptic, so I am on it, and that's possible. But I may have to do more sequencing for cryptic, and if that's the case, then it's 2 months out.
You could say the markers are being a bit cryptic. I'll see myself out. Let's see, another one from Geeked on YouTube. Where are you at with all the other alleles in the 8-ball complex?
So I just happen to have the most of all of the 8-ball complex. I happen to have the most of black pastel and inchy, so that's why I went forward with those. I honestly don't know for sure. I know I have a few cinnamons that have already been sequenced. Obviously, I have several that you all have sent in that I haven't sequenced yet. So yeah, honestly, I'm happy to hit a couple in that complex. We're going to hit 1 or 2 in the black-eyed Lucy complex probably this month, 1 or 2 in the blue-eyed Lucy complex probably this month. So we're kind of hitting some, here and there, not necessarily trying to get all of them within a complex. Dr. Siddell did an awesome job with Yellow Belly. They had a lot of resources and focus and students helping to be able to nail that whole complex. What I'm working from is just what I have the most sheds, and it happened with black pastel and inchy. I had the most of those, and for some of the other complexes, I have 1 or 2 I have by far the most of, so I'm going to hit those first. And then we know a location, so now I know a general location for black pastel and inchy. So I won't need as many cinnamon sheds to find cinnamon. I won't need as many sheds to find some of these others where we've already found some within that same complex. So they'll be easier, but like I said, I'm just working through whatever you all are sending in, whatever I've got the most of, I'm going to nail those first, just because the more we can sequence of a given mutation, the more likely we'll be successful finding it and designing a test that works.
Very good. Actually, kind of related to what you were talking about, are you finding pattern or colored mutations in similar genomic regions, or are they scattered across the genome?
So I've kicked around the idea of putting up a graphic. To me, I love looking at it because I'm definitely a DNA genomics nerd. It makes me happy inside because I've seen so many karyotypes and this, that, and mouse genetics, blah blah blah. But you don't get to see it for snakes. So
Yeah.
I might put one up maybe when we get the monarch information, so we know where the general mapping for monarch is. But yeah, it's really interesting. That's a great question, PhD Pythons. I was actually messaging back and forth with him earlier today.
Yeah.
But yeah, it's pretty cool. It's been amazing. The mutations we've looked at so far, almost every single one has been on a different chromosome. There's only where they've ended up on the same chromosome, but they're like at polar opposite ends, and there's complete linkage disequilibrium or linkage. They're not linked at all. They're independently segregating. I know that he knows these terms, but so yeah, they're far enough apart on the chromosome that we can't tell as breeders that they're on the same chromosome. So yeah, it's been pretty cool to see that. We are getting to where we have enough of them that there are 2 or 3 that are in pairs that are on the same chromosome.
Very cool. I really hope that I'd like to see that graphic myself. Even as a layman when it comes to genetic talk, I'd love to see it. Make it a ball python genome for dummies. I don't know. Let's see. From a YouTube comment from Ray Navedo, are Morph Market and Clutch the only way to order a shed test?
Yes. Yep. For now, that's the only way to order for us to run that for you. And I'll let Sean jump in if you want to add anything to that. But yeah, that's really from my standpoint as the genetics, the scientist, the DNA shed dork that I am. If I had to spend time figuring out how to record results and send out results and all that stuff, it'd slow me way down being able to design new tests and do the things that I'm really good at. So there are only a few things that I can do that I can't really easily teach someone else. And so it's really good for me to focus on the things I can do that other people can't, and let other people that are really good at other things do those other things. So that's why we've had them help us out, because it's something that would slow us way down. We wouldn't have anywhere. I don't know if we'd even have anything available if it wasn't for Clutch and Morph Market helping us out. But is there anything you want to add, Sean?
Yeah, really. Right now, we don't have any plans to add any other partners. The two that we've got have done great, and honestly, Ben and I are both pretty loyal people as far as friendships go. And both of those companies didn't have to take us in, but they did because they recognized how valuable this could be to the industry. One of the things everybody should understand is there's a couple of types of software that are involved in this that we have to manage, and one is the sales and marketing and the side that you all see, and ultimately that's going to lead to lineage tracking for any given animals that not only that you have now but that you've produced in the past. There'll be records of that genetically if you choose to go that route. That's a whole different level of software that when we finally broke the ice on this, Ben and I, about a year ago, we've been chipping away at it for 11 years, and it went from, man, when are we going to get there, to, oh crap, we're here. Now what? And it became appallingly clear that the sales and marketing, when you start looking at it from what can we do with this going forward, it was going to be super complicated. It was going to require teams of programmers, not just a programmer. And we already knew a couple of folks that could help us with that. And so we contacted them, and they both jumped on board. The other software that people don't realize, and what we've been investing in behind the scenes, is actual genomic software, stuff that we can use to track DNA into the future. A lot of this data that folks are sending in, we're running and having sequenced and saving for our own scientific uses. But there are lots of reasons why hobbyists, just as customers and hobbyists, can use this data into the future. They need a place to store that. They need software to translate that data into new test results, hint, hint, things like that to the future, right? That requires actual real database software with programs designed to pull data out, translate them into test results. That's what we've been working on. We are a small company. It's literally just the three of us right now. And it has been since October. Prior to Kayla, it was just the two of us for 10 years.
Yeah.
The software is super important. And from our perspective as the science nerds, we wanted to focus on the genomic software and the genomic programming, and outsource the sales and marketing and, tracking of lineage and stuff to somebody else, because we can't do it all. It's too much all at one time. And honestly, I don't see a reason to do it on our own anytime in the future. Again, none of us would be here, customers included, if we had tried to do this out of our garage on our own. We just didn't have the software to support it. There's not enough manpower to collect the sheds, do the research, do the testing, find the results, update all the customer results, answer all the questions. It's too much for 2 or 3 people to handle. And we needed real partners for that. And that we chose Clutch and Morph Market for that reason.
Mm-hmm.
Sorry, long-winded answer, but the reality is there's 2 ends of the spectrum. When we focused on the behind-the-scenes software, so somebody else could do the marketing tracking.
I think it's good to know that insight, because we don't really get to, and most people don't see that other than just the front-facing part of the business. So,
and me being new to this entrepreneurial kind of stuff, this is probably something Sean's gone through a bunch of times, but to me, what has been really interesting is run into chicken or the egg situations all the freaking time when you're a new business. Like, okay, I have to have these reagents to be able to run these samples. Okay, I need them delivered to me for me to be able to use them. We can only deliver to a business that has a brick and mortar. I don't have a brick and mortar. I'm not going to get to the point where I have a brick and mortar unless you send me these dang reagents and I can run tests for people. So there's all these chicken or egg things that pop up all the time. And I know Sean's, he's gone through this a million times with a bunch of different types of things, but it's things that the other people like me as a customer for businesses that I use their services, I don't think about it all. But yeah, there's a lot of things we've had to figure out. How do we make this work? And there's been similar types of businesses in other industries, like testing sex determination for birds and doing different types of genetic tests for horses and cats and dogs. But like exactly what we're doing, no one's ever done it before. So it's been very interesting.
Very cool.
Sean's just knowingly, wisely
Yeah, I mean, I can't add anything to that. I mean, if anybody was a customer right in the beginning in October when we had so many foibles and bumps and bruises along the way, that's exactly why. We thought we could just order stuff and it would be delivered to our houses and we could run tests out of the garage that we built, turned into a lab. And it turns out, no, you really can't do that.
Oh no.
Lots of little things like that where we just weren't expecting it. We had to build a real lab. We had to invest in real equipment. You know, we had to get a real brick and mortar address to send all these things to. And yeah, it took us a minute, you know. Yeah, absolutely. Are we where we want to be? Nope. We have a long way to go. We have a lot of equipment we want to buy. We have a lot of things that would increase our efficiency, increase how quickly we can get results to customers. But we're just not there yet. And every couple of months, it's another big challenge. Like, okay, do we spend the money on research or do we go buy this machine so that we can cut our wait times by a week? It's that sort of thing.
Yep.
I'm along for the ride. Okay, next question we have also from Critical Constrictors on YouTube. At that point where you may need to put the brakes on ball pythons, would you consider creating or expanding multiple teams that prioritize certain species to help with the overload? I assume that's with like research and stuff and testing.
Yeah, we've certainly talked about, you know, hiring, and this once again comes down to you all that are purchasing tests. If there's enough need, then we can hire another research scientist like myself and double the amount of new tests we can put out. But you know, that's just like these decisions Sean was saying. Are we going to hire another person or are we going to get another machine? Do we want more new tests or do we want to be able to run the ones we have faster? So those are the kind of questions we have to answer. But the more tests people are purchasing, more money that's coming in, and we're just turning around and putting that to research and development, then we have a choice. If we don't have those samples coming in, then we can't do either. But if we do have it coming in, then we can do one or the other. And if we have tons more coming in, you know, summer's coming, babies are coming. If we all of a sudden are getting, you know, 10 times as many samples as we have been, then yeah, we might hire another person or two that can help with the design part. That's the part where you kind of have to have like decades of experience to be able to do a good job that will work properly and have the right kind of quality control to be able to have results you can trust. There aren't very many people that can do that. So,
hand up. Can I speak real quick?
Of course.
So Kayla and Ben, or Kayla probably doesn't know this, but Ben does. We already have somebody else on staff part-time who's helping with prepping sheds. When they come in, I'm not checking that stuff in anymore. I'm too busy with other things. So now we have an office staffer helping check sheds in, pull them, label them, get them prepped for the first few steps before we actually send a plate off to get sequenced. So yeah, I mean, as we grow, obviously we're going to bring more people on. I would ideally like to have another PhD, preferably a younger one. No offense, Ben, to your age.
None taken.
No, there's a lot of really cool stuff coming out right now. And there are a lot of PhDs coming right out of college that are very familiar with technologies that are super impressive, that could really help us with our speed and our cost and things like that. And as a business, we can't ignore that. So at some point, yes, I can see that we'll have probably a couple of PhDs on staff and 2 or 3 team leads that are responsible for different research projects, depending on, maybe a ball python guy, because that's such a big market. And then maybe another person who's responsible for all colubrids, and then another person who's responsible for all other that. Can y'all still hear me? I know I keep timing out a little bit.
Get out just for a second. Other python species.
Other pythons and things like that. I live way out in the country, so I've got crummy internet here. But yeah, I mean, absolutely, that's the direction we're headed. What's going to tell the tale is how fast we're going to get there is the next 18 months. Busy season is almost upon us, and that's why we're pushing as hard as we are with research. We want to deliver as many tests as we can to you guys before all of your babies hatch, hopefully, so we can get an idea of what the next 4 months, when everybody starts getting tons and tons of babies, is going to look like from a revenue standpoint. And then we can plan better, right? If we know to expect X number of dollars off of the 18 tests that we have available, if we have 36 available this time next year, I can make some sort of a reasonable guess on what our revenue should be, and we can plan for that. And staffing is the biggest part of all of that. So yes, but it depends on you guys. Send us your sheds, please. Send us your sheds. And, on another note, by the way, it's not going to happen this year, but I know a lot of you guys are waiting for quantity discounts. We do have that in the works, but it's not going to happen in the first year. It's just not. We've made that not only this decision as a company, but we talked to both of our vendors, and they sort of agree that this first year, let's get a feel for what 1 year looks like. And then next year, maybe we break up some panel tests. We lower some pricing. We have, again, another product offering that we want to bring to market, which will probably be our top end product at that point. And it's going to be a complete pricing kind of structure change. So I understand I'm probably talking myself out of some sales, but please send us your sheds right now. And I promise you this coming year, it will pay off in spades. The prices will come down for quantity. We'll have twice as many, if not 3 times as many more tests, excuse me, for ball pythons. And that might actually be a good segue, Ben, for you to talk about, what's in the works right now. What can we expect in the next 6 weeks? Because I know there's a bunch.
Yeah. Partly related comment, like, spot nose and GHI.
Yeah, so spot nose and that spot nose, spider, blackhead, all those. I do have some data already on those. I do know what chromosome they're on, but I don't have a test for any of them yet. But that is a complex that I should be able to pull out 1 or 2 within the next 2 to 4 months. That specifically will be a little difficult. GHI is kind of in that same boat. I have enough sheds that I do have some sequence data for GHI, but I have not. I don't have good enough candidates to start chasing yet. But that is another one that's decently close. I don't know. We're an hour in. Do you think I should talk about the registry one? Should I crack that right now, or?
Yeah, I mean, I guess we had talked about releasing it in the future, but I've already referenced it a couple times, so you can at least kind of drop the hint. It's going to be a couple months probably before we can actually make it available through our vendors, because again, it takes some programming. But I mean, yeah, you can tell them some of the things that we're capable of doing now and why we think it's probably going to replace a panel in the future.
Yeah, so the really cool thing with this, so that this is probably going to, we'll call it something like the genomic registry or something like that. What we'll be able to do for a pretty reasonable price, it'll be a little higher. It'll certainly be higher than the panel test right now, but I think, correct me if I'm wrong, Sean, but I think we'll be able to keep it under $200, and it will definitely be a little high. Well, maybe not much higher, but it will be higher than the panel test right now. But what this would allow you to do, which is really cool, I'm very excited about this. We will be able to take your shed, and for this genomic registry price, we will be able to use this genomic software that we've been developing and be able to save whole genome information on your animal. So all the time, and actually I can see some of the questions I think that Kayla even has highlighted, if we can get to them tonight. You all have tons of questions. They're really good questions. Really, really cool. I'm glad there's other genetics dorks and nerds and addicts out there besides just Sean and Kayla and I.
Yeah.
But yeah, what this will allow you to do, lots of people ask, hey, I sent my shed in for het clown test, and now you just came out with hypo. Can you run it for hypo for me? Or can you get that information from, and, we have to do that all over again. We have to extract the DNA again. We have to run the test again, unless, we just did it last week and I happen to still have the DNA in the fridge. But what this genomic registry will allow us to do is we would be able to, once you pay for that genomic registry, every time we come out with a new test, you can access that information about your animal, because we're sequencing everything. And so even though we don't know for sure where monsoon is right now, if we get that in 4 months and you've paid for that registry, then I don't know for sure how the price structuring will be. Maybe it'll be $25 to run the new test. You're essentially just paying for us to go in and pull that information that's already there. We just didn't know which of that information was important. Mm-hmm. And so, yeah, exactly like this question right here. If you pay for a full panel before you release the 2 genetics, DG and genetic stripe, and I haven't shipped the shed yet, will DG and GS be included? So in the case of a genomic registry test, then yeah, you would be, for the whole rest of forever, anytime we come up with a new test, you would be able to access that information. 5 years from now, if we have markers that show us females that will give you larger clutches, which that's something we'll definitely be able to do over time. It will take a few years, but we could say, hey, yeah, this female has 3 of the 5 markers that show us she's going to give large clutches. So over time, there'll be more and more information. And once we've taken that genomic registry, once we have that information saved for you, every time something new comes out, we'll be able to let you know your animal, its information for that new test. And so you won't have to send in another shed. You won't have to, it's no new thing. We would go back to that sequence that we already have for that animal and analyze that for your specific animal. And so it's a lot more like, for me specifically, I can't remember if it's ancestry.com or something like that, but they'll send me an email once a month and say, hey, one of your relatives, just send in their sample, blah, 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 4th cousin, whatever. Or I had one just recently say, hey, this specific cemetery in Utah, you have so many relatives there, whatever. So we'll be able to tell stories. We'll be able to say this animal is, offspring from one of, Bob Clark's original, blah, blah, blah. Like, we'll be able to track that kind of stuff over time. Sean, sorry,
yeah, you were kind of hitting on it. The cool thing about it is we're going to have a genetic fingerprint of that individual animal, and pricing is going to be bounced around back and forth. And we're taking feedback from you guys too. It's going to be more expensive if you want to pay less for tests as they get developed later, or it's going to be less expensive if you're willing to pay a little bit more for new tests as they come out. The best thing about it is just that once we have that genome for that particular animal, we basically have the genetic fingerprint of your animal. We can pull test results out of it forever at that point without you having to send a shed in. And honestly, you'll get a result within a day generally. We just have to receive your order, run the software, let the program work its magic, figure out a result, and send it back to you or update it into the system. But to touch more on what Ben was saying, one of the things I like the most about it is because it's a genetic fingerprint, lineage tracking is now a thing. If you have a line of particular animals, I know over the years people have had pastel lines and different lines where they're just seeing, this is my particular line of animals, whether it's line bred or not. If you have a founding animal, we can register that animal, and every offspring over multiple generations, we'll be able to tell you if it's related to that original animal. So it has a lot of potential power. There's if you're one of those folks that has a big collection and it's insured, the ability to have fingerprints on those animals, so that if they're ever stolen or anything like that, is a real thing. It can lower your interest rate. So there's a whole lot of different ways that this can be used besides just genetic testing. So
that's huge.
Question, and that's, again, we're probably a couple months out before our marketing partners figure out how they want to offer it on their systems. And then, how do we want to pitch it to you guys? I mean, did we do the high price, but then tests are free forever into the future, or do we do a more reasonable price and, hey, the next time a test comes out, send me $10 and we'll give you a result for the new test?
Yes. And another question similar to Alicia's question earlier. I purchased a full panel test. We also add DG and GS. I haven't sent the shed yet. So what we were just talking about, that's 2 or 3 months from now, you'd be able to do this genomic registry, and forever after that, whenever we have a new test, you can find out if your animal has that gene or not. But right now, so to answer the question right now, if you paid for a panel, you haven't sent the shed in yet, and Sean will be the man to set you up, hook you up with this, just right on there, say, please run DG and GS, and we'll mark it as such and make sure that we get those run for you. If there's a mistake, just let us know. When you get your results, say, hey, I really wanted to get DG and GS, and we'll try to make it happen. Since it's not in the system, we might miss one here or there, but Sean's been really good about marking those. And then when I get it here, I make sure I get those added in so that you can get those results too. We're happy to do it. Just make sure you write really clear. Just say DG and GS, please.
At some point, though, that's going to have to stop. Right now, it's not a big deal. But the reality is that if you've been with us since the beginning, Ben let you in on a little secret that we have our little Dewey Decimal system of thousands that you guys have been sending in. The problem with that is as that library of sheds grows, it gets harder and harder for us to pull a shed if you've already sent it in and we've already, pulled it, prepped it, and sent it in. for results. So a lot of folks, what's happening is they're getting results, or right before they get results, but after we've sent their shed in, they're asking us to add stuff. And the only way we can do that is to go back into the pile, pull it, and reprocess. And that adds a lot of time and a lot of cost for us. Right now, we're trying to help everybody as much as we can because we're trying to, make a good impression on you guys. But at some point, we're going to get to the size that's going to become too time consuming. So right now, yes, if you purchase a full panel, especially if I don't already have your sheds, just right on the bag, we'll make sure it's prepped the right way. But if I've already got the shed and it's already been prepped, or worse, it's already been sent off for sequencing, when you ask me to do that, I have to dig your shed out of an old box and redo everything. And that's pretty expensive for us to do. So
and
keep that in mind as we're trying to adapt. However, the bigger we get, the less customizable our service is going to be when it comes from that sort of an angle.
Yeah.
That's why it pays to get in early, at least.
Yeah.
And honestly, to let you in on another little secret, your shed gets filed in a way that we can find it faster if you're telling us what all genes are there.
Right.
So if you're letting us know that, then it's more likely I can find it or Sean can find it. If it's one where you don't give us any information and it's already been processed and run, and then you say, oh, can you add DG to it? Then the chances are very high that you're going to have to send another shed in because
we've already thrown it away to save space. Yeah.
So it's another reason to give us more information, because your shed will mean more to us. We'll save it and keep track of it better if we know there are other genes there that we can test for and things like that.
Yeah. We have a question from Olivia Wright. You mentioned doing something with spider before, but she mentioned it'd be interesting from a diagnostic sense for animals that display neuro issues or just basic wobbling.
Yeah, I'm excited to work on that complex. That will be an interesting. I see that being a pretty cool paper. I don't know if Olivia, if you have seen it, but there is a really interesting paper on the spider ball python morph, and specifically physiologically what's going on. It's really cool. They have I think there's some really cool microscopy in it and stuff like that. Even if you're not a science paper reading person generally, there's some pictures and general information there. If you just read like the abstract stuff like that kind of give you a better idea. But as far as the actual mutations that are causing spider and the other morphs in that complex, yes, I'm very excited to learn from that and write up a cool paper about what those genes are and what's going on. And that might be a project that if it's something that Dr. Seidel is interested in working on, we may collaborate with her on that, because she's got that academic side, and that's hard for us to match. We have a very different focus than she does, but from an academic standpoint, that's a very cool complex that will be really neat to learn what the genes are and what's going on, and I'm excited to learn that.
Well, and it's one of those things that you brought up earlier, Ben, too, that's a mutation that's actually present in several other species that you can find exactly the same sort of things going on. So it's probably pretty much exactly the same thing, if not exactly the same thing.
Yeah.
That can all go into the same paper. So yeah, that one's super interesting. We've talked about that a few times.
Yep. Yeah. As a grad student, there was a chicken geneticist that was in the same department as me, and we would talk about animals all the time. And I brought up spider ball pythons. He's like, oh yeah, there's very good work done in poultry. Very similar, the mutation that changes where the dark pigment cells go also changes your neural connections. And so yeah, even in chickens, but blood pythons, there's the batik. In carpet pythons, there's the jaguar. Those I would imagine.
Jaguar retics are probably the same thing.
Yeah. Oh wow! Wait, so you're telling me there are spider chickens?
Yep.
Excellent. We need this. This needs to be. We were talking about maybe doing a spider genetics episode at some point, maybe. So that would be cool. Yeah. All right. Awesome. Speaking of, we mentioned retics actually. Joe Ceballos, I think is the appropriate way to pronounce that. Any retic tests soon?
That depends on y'all. I've had several people talk about sending retic sheds to me, but I don't remember getting any. If I have, it's only a couple. I have gotten some Burmese python sheds. I got a blood python shed. I'm starting to get some other python species, but I really don't have very many retics at all. So you can talk to people. Actually, Kayla, you said you were messaging with Bob Clark the other day, and he showed some interest, right?
Yeah, I've been talking with him. He says he's going to start saving up sheds and sending them. But any retic people people who have them, I don't know, an interested zookeeper, I don't know. Those sheds would be great.
Well, we are willing to travel also, by the way. If you have a big enough collection and we can knock a lot of birds out of the air with one stone, well, we're willing to come to you and do scale clippings and all sorts of other DNA extraction methods to try and get what we need to start developing tests. But yeah, the retic folks, you guys are great. I have a couple of retics myself. However, y'all aren't doing a great job of sending us sheds. So please send us what you got. If we want to work on them. You guys have so many wicked cool mutations, and we've got maybe 30 sheds across all of them.
Wow. So pump up those numbers.
Yep.
We have one from Geeked. With Desert Ghost available, did you know if Enhancer turned out to be allelic or the same yet?
So I've only run probably 5, 6, 7 that breeders had marked as Enhancer, and the mutation I'm seeing so far looks identical to what I'm seeing in Desert Ghosts. But that being said, I don't have it fully figured out. So that's one. That was one of the 5 I was hoping we would have completely nailed down this week. So Cryptic, I got no information on. Desert Ghost, we did get some really cool information, and we understand it much better. And it's going to be very, very cool. It's going to be a very
That's going to get its own show.
That's going to get quite a bit of information and education about. It's another one that is definitely not as simple as we thought it was. And any of you that have worked with Desert Ghosts for several years and produced several clutches, once we explain this, I think you're going to have a lot of aha moments from clutches you've had in the past and questions you've had. And it's pretty cool. It's much more complicated than we thought, and we will get all of our ducks in a row and definitely share that information. The test that's available now on Clutch and potentially could be offered by Morph Market very soon.
You can still order it through Morph Market. You just have to order any other test that you know your animal doesn't have and just write, please test for DG instead of albino or whatever test it was that you ordered, and we'll run it for you.
Yeah, we'll run it. But yeah, Clutch is marketing it as a elimination test. So if you get a negative, that's 97% chance it's really negative. If you get a positive, it's 85% chance that it is actually a het. So it's really the strongest is if you want to eliminate animals, you run them, find out which ones are negative, go ahead and sell those as not being het, because most likely 97% chance they're not. The ones that test positive or het for DG, you can think of those as an 85% pos het. So it's helpful, but it's not as good as our other tests. And like I said, Enhancer so far, to me, I don't see any difference, but I've only looked at a few. So once I've looked at 20 or 30, I'll be able to answer that question in more depth. But so far, it looks the same.
To be determined then. This one's from Morph Valley Reptiles. When is azanthic testing going to be available? VPI, TSK, etc., to confirm which is in a collection.
So VPI and TSK, we mentioned a little bit earlier, those two will be available most likely sometime this summer. So it could be as fast as 2 months from now. So we're kind of in the middle to end of the summer will likely be when the TSK and VPI will be available. The other azanthics, I just don't have enough sheds. VPI and TSK are the most common, and I have by far the most sheds from those two. So if you're interested in any of the others, you just got to get sheds to me. And like I said, we're making a priority to put research and development dollars towards that right now. So this is a good time to get sheds to us.
All right. This one's from Clover's Reptiles, and I think it's our second to last question. For Western hognoses, do you need sheds from single gene snakes, or can it be from multi-gene snakes? And do they have to be homozygous?
The answer is yes. We want all of it. So if you
Send
have
them all.
if you have single genes, we want it. If you have multi-genes, we want it. So if you have a single gene, we can use that for that single gene, and we can also use it as a negative to compare any other gene to. So if you have an albino, and for whenever we develop tests for lavender albino, conda, all these others, we can use that as a negative to compare to. So actually having normals or ones that just have one gene is helpful. The multi-gene ones are helpful because if you have 4 different genes in it, when we're developing those 4, we can use that shed for all 4 of those tests to help us develop it. So they're really equally valuable to us, whether they're a normal, a 1 or a 2 gene, or if there's a 4, 5, 6 gene, all of those are valuable to us. But they have to be labeled correctly. So if you send us a shed that's 2 different morphs and you label it correctly, that's awesome. If you send us a shed that's 2 different morphs, but you tell us 4 different and we start to try to use it for other things, you'll slow down our ability to get those tests done.
Offsets are not helpful either.
No offsets. Yeah. I mean, you can send in an animal that's a multi-gene and posset for something and definitely tell us that, so that we don't, like if it's posset for albino, I don't want to use that as a negative to compare to because it might be albino. So definitely let us know if it's a posset for something. So we want to know an animal name or number. We want to know any genes it's homozygous or heterozygous for, whether it's recessive or incomplete dominant. Doesn't matter. Tell us what you know it has. And then thirdly, tell us what it possibly has, whether it's homozygous, heterozygous, incomplete dominant, recessive, just if it's possibly there, let us know. And then sex is also good to let us know. So those 4 things on every sample you send in, make sure we have at least those 4. That would be perfect.
Yeah. And related, what about snakes where you don't know what the genes are? In this case, Zephyr York Munkers, please don't hate me for mispronouncing that. I've got an albino boa. Would you all benefit from me sending in one of his sheds to use if I don't know anything else about it?
Yep. Yeah, that's great. Send it in. Albino boa is especially interesting. We did an episode, I think it might have been episode 4, something like that, that was specifically on the albino conundrum that's going on in boa, because early on, the 2 different strains, Sharp and Call, were bred together, probably in a lot of cases unknowingly, and then sometimes even knowingly. And so it's hard to know for sure if you have an albino that's just a Call or just a Sharp. So we would love to untangle that and be able to have a Sharp test and a Call test, and that way people could have pure Calls and pure Sharps if they wanted to. But that's really only going to be possible with genetic testing. So yeah, send it in and we will definitely use it.
Very good. All right. I think that's all the questions we have from our commenters today. And we've still got almost an hour and a half in, and we still have 32 of y'all. You guys are the best. And that's not even counting Instagram. So
yeah, on Instagram, we've had between 10 and 15 most of the time.
All right. Very good. Well, you guys got any final thoughts for today? Anything?
Just a sincere thanks again. Like we talked about earlier, this is what keeps my family going so that I can think about just reptile genetics all the time. Otherwise, I'd have to find another job and go back to doing this part-time. So thank you so much for using our services, for the support, for the good posts and comments and questions. It means a lot personally to me and to my family. Keep it coming.
Definitely. Sean, you got anything for us?
Everybody, please keep Dr. Ben employed. Send us as many as you can when you can. I promise you, as the business manager, I am working on finding ways to get all this done cheaper for everybody, but that's just a matter of us hitting certain milestones. So don't give up on us. Please keep sending us what you've got. You've got my word. Prices are going to change. We just got to hit those milestones first. As you've probably learned today, we dropped a few items we didn't intend to drop, like the registry, the DNA registry. There's some really cool stuff coming, right? And that's all because of you guys. So the more of this that we do, the more sheds that we're processing, the quicker we can develop these new product ideas, the quicker we can change the equipment that we own, which will affect wait times and prices and things like that. So keep sending us what you got. I recognize it's a little bit expensive. I wouldn't want to pay some of these prices either. I really believe that, but we need you to right now. We really do. we're doing really well, but we have so much more that we want to do. So we haven't even really cracked the surface. We're finally starting to talk about a few of the things, of the hundreds of things that Ben and I have been talking about since 2011. So thank you guys very much for your support. And that's all I've got.
Yeah, thank you guys. Make sure to like, comment, and subscribe. And also much love to everyone who watches this video on the replay. It's still going to be up on YouTube and Facebook for you guys to see. So thank you. And with that, I think we'll call that a wrap on live stream number 2 and episode number 9, I think, something like that. Anyway,
Yep.
Thanks, guys.
Thank you.
Outro in 3, 2, 1.
Feel free to watch it on Instagram or Facebook. Then you can see their faces too. But with that, I'll be out on Instagram.
There we go.